Good afternoon, friends. Myself, Sangmeshwar Shivsam Abande. Today I am talking about the restriction enzyme. See here, restriction enzyme. What are they? Restriction enzymes are enzymes that have been found in bacteria or invented in a lab. Restriction enzymes are used to break apart restriction DNA into individual fragments or genes. Then next, how do they work? Restriction enzymes are actually enzymes created by bacteria which the bacteria used to defend itself against DNA by breaking down and foreign DNA it come in contact with. Scientists Werner Erber and Stuart Lin found the specific section of DNA that did this and named it ECOB. This is the first name restriction enzyme. Then we will see next how much do they cost. Restriction enzymes can be bought on the internet. Their prices run from S20 for 1000 units to 500 for 1000 units. Price based on demand for the enzyme in research. Then next, type of restriction enzyme. We will see one by one type. First type is enzyme recognize DNA sequences but cut the DNA in random sites that can be as far as 1000 base pairs away from the recognized site. Type second enzyme recognize and cut within the recognized site. Then type third enzymes recognize sequence but cut at different location within 25 base pairs of the recognized site. Then next, subunits of type first restriction endonucleases. Type first restriction enzymes possess three subunits. HSDR is required for restriction, HSDM necessary for adding methyl group to host DNA methyltransferase activity. HSDS important for specify of cut site recognition in addition to its methyltransferase activity. Then second, type second restriction endonuclease. The first type second enzyme to be isolated was Hind second in 1970. Only type second restriction endonucleases are used for restriction mapping and gene cloning in view of their cleavage only at specific site. Then next, subunit of type second restriction endonuclease. These subgroups are defined using a letter suffix. Type second B restriction enzyme, type second E restriction endonucleases, type second M restriction endonuclease, type second restriction enzymes. Cut off type third second restriction endonuclease. Type second restriction enzymes can generate two different types of cut depending on whether they cut both strands at the center of the recognition sequence. The former cut will generate blunt ends with no nucleotide over changes. The latter generates sticky or cohesive ends. Type second restriction endonuclease. Third restriction enzymes are intermediate between types first and type second endonucleases. Then cleave DNA in the immediate vicinity of their recognition sites. Example ECOP1, ECOP15, Hindus third, etc. They cut DNA up to 20 to 30 base pairs away from the recognition site. Three enzymes contains more than one subunit, ATP and magnesium 2 plus, and require ADO, mate, and ATP cofactors for their roles in DNA methylation and restriction. Recognition sequence of RS enzyme. The recognition sequence of type second endonucleases from palindrome with the rotational symmetry. Mostly type second endonucleases have recognition sites of 4, 5 or 6 BP which are predominantly CG rich. Thank you.